Hello everyone, my name is Nathaniel Thomas and I am an undergraduate senior at the College of Science in the University of Utah. I'd like to thank the Office of Undergraduate Research for giving me this opportunity to present my research, as well as the Planelles Lab for giving me the opportunity to conduct research. Through them, I was able to learn about IFITM3, SAMHD1, and the interferon response to viruses. HIV is the disease that is focused on primarily at the Planelles Lab, and HIV is a very interesting disease. The first few cases were reported in 1983 to 1984, and despite being discovered this early, there has been no preventative vaccine developed against it. Around 37.9 million people have HIV worldwide today, and there's around 1.7 million new cases of HIV reported each year. Now, despite these discouraging statistics, HIV has been very important and instrumental for a few scientific discoveries, especially those relating to gene therapy through vector technology. Now, gene therapy did exist prior to HIV, but there were some limitations and some problems with gene therapy. And HIV was important because it allowed researchers and scientists to infect non-dividing and terminally differentiated cells that were previously unable to be infected through the vectors that were used. HIV completely changed this and is still used in a few clinical settings and quite a lot of experimental settings today for this reason. A few other important discoveries of HIV were the different proteins that have been found to restrict the growth of this virus. A couple of these include IFITM3 and SAMHD1, and IFITM3 being the interferon-induced transmembrane protein 3, and SAMHD1 being the sterile alpha motif domain and histidine aspartate domain containing protein 1. These were, and still are, two important proteins that are looked at quite often today, and the Planelles Lab decided to focus quite a bit on these two different proteins and their relationship with HIV. One of the most focused proteins that we looked at was SAMHD1. And SAMHD1 is especially interesting for a few different reasons. SAMHD1 is phosphorylated on threonine 592, or T592, which turns off the effector function of SAMHD1. This means that if SAMHD1 is not phosphorylated, it will cleave deoxynucleotide triphosphates, or DNTPs, into their nucleotide base and inorganic phosphate components, as seen in this little graphic here. This will decrease the level of cellular DNTPs, which will prevent reverse transcriptase from occurring after HIV enters the cell. From these observations, it can be determined that if macrophages are infected with HIV, the infection process itself will dephosphorylate SAMHD1. These were very important discoveries in the field of HIV research, and this left people in the lab that I'm working at wondering a few different things. And one of the most interesting questions that was asked was, does infection with HIV result in a protective bystander effect? Or in other words, is the initial infection of HIV causing a protective bystander effect that will protect these cells from a secondary infection? And these cells that we were looking at, to clarify, were macrophages. So experiments were done to test this hypothesis, and they looked at varying levels of an initial infection of HIV, which was stained with M-cherry, a red fluorescent protein, while using a consistent level of a secondary infection of GFP, the green fluorescent protein. And this can be seen here, the different varying levels of primary infection ranging from 0 nanograms all the way up to 100 nanograms with 5, 10, 25, and 50 nanograms in between. And by analyzing the two different colors of fluorescent proteins through a flow cytometry, we were able to find these results over here that clearly showed that there was some sort of bystander effect or protective bystander effect occurring. As can be seen at 0 nanograms, so no primary infection, there was around 23.5% of cells being infected, which makes sense. And then once we reached 5 nanograms, there was a clear major drop to around 10% infection. This can be seen as you keep going down, and at 100 nanograms of the primary infection, there was around a 0.04% secondary infection rate. This was a very clear indication that the bystander effect was occurring and that HIV was kind of preventing a secondary infection from occurring. So this was very interesting in our lab, and this is when I entered the picture in the lab itself, and where I was wondering, and as well as other graduate researchers were wondering, what exactly was causing this bystander effect to occur. Now, there were many things we could have tested, but the one that looked the most promising towards us was interchanging the viral envelope, being the HIV envelope itself, and seeing whether this was causing the bystander effect. This is what looked to be the most likely to us, which is why we tested this. And how we tested this was by creating pseudotyped viruses. So we had different virus envelopes around a same core. So the different virus envelopes, I'll explain this. The different virus envelopes were HIV with the subtype JRFL, VSVG, AMLV, GMTR, Lassa virus, RD114, and LCMV. So many different virus envelopes here, trying to determine whether this was what was causing the bystander effect to occur, or if either any of these envelopes would increase it, if they would hurt it, whatever the case would be. And we maintained the same HIV core because we didn't want to have too many conflicting variables, and at the time it didn't seem like this was the most likely thing that was affecting the bystander effect. Now, how we went about this process was very interesting. There were many different methods involved in this process, and they were conducted, some of them at the same time, but I'll split them up here for you. So we would do bacterial transformations, cell splitting, and viral transfections to create and allow for this experiment to occur. To conduct the transformations, we would take plasmids and grow them on an agarose plate, and these plasmids would have an antibiotic resistance gene. We would then pick these and then put them into a culture of E. coli, and this E. coli would grow, and hopefully some of it would take it in. Then we would plate this E. coli on an antibiotic resistance agarose plate to conduct a maxi prep and select for the viral plasmids. The next step of this process was splitting cells, and how this would work was, well, we would thaw human embryonic kidney 293F T cells, or HEC 293 cells, and we would let them sit for around 3 to 5 days because they had been frozen, and after these days of thawing, we would split the cells so that more cells could grow, and also so that they would have enough resources to grow in general. So we would continue splitting the cells every 2 to 3 days to make sure we had enough flasks of cells to make enough virus. And this leads us to the viral transfection process, where we are combining the two different parts here. And to do this process, we would use polyethylenamine, or PEI, as well as lipofectamine 3000, which is a reagent that is used often in the transfection process. Now, these are important for transfection as they allow for the plasmids that were used in the transformation process to enter these cells, the 293F T cells, and this allows for replication to occur in the cells, and then this ultimately allows for the virus to leave these cells and enter the media. The media would then be collected, frozen for storage, and then thawed for experiments later on. Once we had our donors, we were able to then carry out the final stage of the experiment process. So donors, we would have blood donors who would give 120 to 180 milliliters of blood, and we would then centrifuge this blood from them and collect the peripheral blood mononuclear cells, or PBMCs, from them. PBMCs themselves contain lymphocytes as well as monocytes, and we are specifically interested in the monocytes because the monocytes are the ones that differentiate into macrophages and dendritic cells. So after centrifuging and collecting the PBMCs, we would isolate the CD14 monocytes and then differentiate these into monocyte-derived macrophages over 5 days in pooled human serum. On the 6th day, we would usually change the media to allow for better growth. And on day 7, we would finally conduct our first infection. And on the 8th day, we would conduct the second infection. On day 9, we would usually change the media again to once again allow for better growth. And finally, on day 10, we would stain the cells to look at the flow cytometry to determine whether the cells are viable, meaning whether they're live or they're dead, as well as the level of infection, which was determined by whether they were red or green, red being M-cherry and green, of course, being green fluorescent protein. Now, instead of showing all the flow cytometry plots that we got, I thought it would be a lot easier to visualize all of the things that we looked at through a table that was consolidating all the information in one space, because there were quite a lot of flow cytometry plots that were made. Now, if you look at this table, you see a lot of these cells are either gray or highlighted yellow. And for the gray cells, we can see that on the far left column, that it starts with 0.44 with JRFL, 0.14 GNTR, loss at 0.22, and RD114 at 0.32. These are the initial infections with the first primary infection using the GFP stained virus. Now, these are colored gray because they're very low levels of infection, and we didn't think that the infection level was really enough to determine too much from the information from them. So unfortunately, we couldn't really look at those, and all the gray cells was data that we really couldn't use, which was unfortunate, of course. But the data that was collected was very useful and important in its own way. Now, before I go into too much depth on the table itself, if we look here, the first row here, the one with second M-cherry, says 0.21, 33.4, etc., etc. It continues on. These are the secondary infection, and just wanted that to be clear, which is why this darker line is here, to kind of separate the two different parts. Now, if we look at the infections, if we look at VSVG, the primary infection of VSVG had a 41%, around a 41% level of infection, and the secondary infection of VSVG had only 6.6% infection rate. Now, this is very, very expected. They were both VSVG, but again, to kind of clarify and make things a little bit more clear, this means that around 93% of the cells were protected from VSVG if only 6% were infected. This was especially interesting when we look at AMLV. So again, VSVG being the primary infection at 41.7%, the secondary infection of AMLV, only 0.12% of cells were infected, which meant AMLV being the secondary infection offered a 99% rate of protection. And this was also seen with LCMV, showing a very similar number as the AMLV. So this was very interesting because we could see that there was definitely some sort of relationship here. Now, if we look at AMLV, it had around a 5% level of infection, and if we look at AMLV with the secondary infection, it had around 2% of an infection level, which meant, again, like 98% was around protected, which again, somewhat expected because they were both AMLV. And then if we look at LCMV, again, had a higher rate of protection, which was very fascinating. But what seemed to be even more interesting in this case was VSVG. This time, instead of offering protection, VSVG actually offered less protection. In fact, more VSVG were infected in this case. And this was a similar pattern that could be seen with VSVG in the LCMV infection, primary infection. So these were very interesting numbers to look at for sure, and they definitely showed some sort of relationship with the viral envelopes, especially the relationship between VSVG, AMLV, and HIV. Now, these were all very interesting, and they bring us into the future plans of this project. So we were looking at SAMHD1 quite a bit, and another thing we were planning and we are very interested in is IFITM3. because we actually do believe IFITM3 may be playing a major role in this viral envelope protection that could be occurring. So our plans are to, after isolating the CD14 monocytes in the process of developing this experiment, we could use neon electroporation to CRISPR out the IFITM3 to see if this is going to completely stop the bystander effect or if it's not causing it. We'll have to see what occurs. Another topic that we've looked at is perhaps changing the cores. So instead of keeping the cores the same, we could change the cores and keep the envelope the same. We don't believe this will have an impact, but we are not sure, of course, and we'll have to see. We'll look at the cores being DHIV and MOV. Those are the two cores we are planning to look at. And finally, the initial infection. As you could see from our results page, they had very differing initial infections, VSVG being 41%, AMLV infection, and LCMV being around 5% levels of infection. So if we are able to control for this amount of initial infection, that would really help, because that could also be why the level of VSVG was so different in this AMLV secondary infection as well as the LCMV secondary infection. Perhaps there just simply wasn't enough of this initial infection for VSVG to actually have this bystander effect occur. So these are the things that we're planning to look at. I'd like to thank the Department of Pathology for giving me the opportunity to work with them, for Vicente Planelles for giving me the opportunity to work in his lab, and the graduate student Elizabeth Williams for giving me so much of her time, so much patience, and teaching me the ropes on everything that I know today that is related to research. Thank you all so much, and thank you all for listening.